This is a production of Cornell University.
I'm going to be talking about cutting-edge technologies in the imaging facility. So as you can see, all the labels that are new are instruments that have just arrived this year or are we're planning to get in the near future. So this is sort of a services overview. We have standard microscopes and fluorescence tools, so fluorimeters, spectrometers. We have advanced microscopies, what we call advanced microscopies, so confocal, multiphoton. We're expecting a super resolution system in January. We have a laser capture micro dissection system, a fax, a CT, and a bunch of technologies that we call intervital imaging, and then support for image analysis and visualization. And so how we normally work is, you make an appointment, we sit down, we talk about what you want to do, and we try and put you on the tool that will work out best for you. If you know what tool you want to use, we'll get you trained in on it, and we generally charge by the hour. But the fax and the CT are full service. So you drop off your specimens and we image them for you. And of course, our staff is our most important resource. Mark Riccio runs the CT. He's going to be tag teaming with me. Carol Bales, right here, runs microscopy and fax. Joanna Delacruz, microscopy and the intervital imaging, and Teresa Pori does CT, super resolution, and some 3D printing. And I'm tangled on the system here. Use that arrow. So our confocal microscopes serve probably the biggest number of users. As you probably know, confocal microscopy was developed for imaging into thicker or cloudy specimens. I do have, no, that's not a laser pointer. It enables you to get a nice, clear, detailed section into what would appear, optically blurry on a normal microscope. So that's really, it's more kind of a one-stop shop now. Our confocal microscopes are incredibly well engineered and intuitive, and people use them for thin samples and thick samples. Basically, you can choose whatever excitation line you need, your emission color, the bandwidth of your emission color, and depending on what system you can use, you can have up to 34 different colors. They have automated stages, so if you want to image something with a field of view that is larger than what your objective provides, it will automatically tile for you, or you can look at different positions and follow them over time. They have automated focus, and of course they have everything to keep your cells happy on the stage. Temperature modulation or gas modulation if you need that. And this is our spinning disc system. This is for if you need speed on the order of 100 images per second. It also has a fast focus motor if you need that. And this system is also good if you're having phototoxicity issues. So this system is supposed to be gentler on your specimens. And now, so multiphoton microscopy was developed here a couple of decades ago in the Webb lab here at Cornell. There's always been lots of user-built multiphoton systems around, not entirely user-friendly. Now we actually, as of last month, we have a lovely, nicely engineered multiphoton system that's easy to use. So all you have to do is type in the wavelength and press go. So it's actually easier to use than some of the standard linear lasers. So what multiphoton gives you is deeper imaging. It's good for thicker pieces of tissue or live plants, live mice, that sort of thing. It gives you access to second harmonic generation and other nonlinear signals. I'm not going to talk about that. Also, probably most importantly, it gives you axially restricted photo damage and photo activation, and I'll talk a little bit about that. So multiphoton microscopy was really heavily adopted into the neuroscience community. This is an example imaging, probably the world record, imaging a millimeter down into the vasculature of mice. Now, especially young brain is pretty transparent. Probably this is kind of a special thing. Most people get around 500 or 600 microns in. But this is an example of some images that have just been taken recently on our new LSM 780 multiphoton system. So this is a cleared poplar leaf. And I can't see it very well from over here, but you can essentially image all the way through the leaf into the, from, I'm going to get, I don't know, leaf terminology, the palisades into the mesophyll here. But you essentially get all the way through. And this was taken by Monica Carvalho from Carl Nicholas's lab. And they're interested in the veinlets here. So this is a single image taken out of this stack right here, and you can see that you can image pretty well all the way through the leaf, the cleared leaf, if you need to do that. So multiphoton also gives you 3D resolved photobleaching, photodestruction, and photoactivation. So I want to talk a little bit about this. So this, if I focus a standard laser beam through a fluorescent sample right here, you see that you get pretty much just as much photoactivation all along the line. Confocal, what it does is basically just image this part, it throws out this part and this part by using a detector aperture that is optically conjugate or confocal. How multiphoton works is it actually just activates at the focal volume right here. So this is a zoom up of this region. And this enables you to do things that are a little more quantitative. For example, if you want to know about the mobility in your sample, you may want to do a standard photobleaching analysis. So in 3 dimensions, it turns out that that's very difficult with normal linear excitation because so you would go along right here, you would park your laser beam, and you get fluorescence. You have a bleaching pulse right here, which bleaches down, and this recovery is due to unbleached molecules that diffuse back into your focal volume. So this is dependent both on your diffusion coefficient and your focal volume. Single photon, it's a not very well defined focal volume. 2 photon, it's very well defined. You can get good numbers out of this. Single photon, you can't get good numbers in 3 dimensions. So this is also applicable to photoactivated GFP molecules such as amyos. So in this case, you excite with purple light and you turn the green molecule to red, and you can watch this diffuse away, and you can quantitatively determine what the diffusion coefficient is. So for mobility. You also, for instance, targeted ablation, if you want to do that. This is an example from the Lammerding lab from Philip Iserman. What they would like to do is sever the actin filaments within a cell without affecting the nucleus. So right here, this is using normal confocal microscopy to image, and they're going to use multiphoton to sever the actin filaments, which are above the nucleus in this case. So you can see that they do that without damaging the nucleus. And this is the 2nd place winner in our image contest this year. So get your images ready for spring. And I just wanted to harp on this for a little bit. This area right now of optogenetics is really hot right now. Being able to optically activate signaling molecules, and you all probably know more about these phytochrome B and as a light sensing protein because it came from plants. And so the idea here is that if you activate phytochrome B at 650 nanometers, it will bind this phytochrome interaction factor, or the blue triangle right here. And people have now engineered this into mammalian systems, where if you optically activate this protein, it will bring a row effector molecule to the membrane surface and basically activate it. And this is an example of that. Microscope PowerPoint has stopped working. Very interesting. Okay, we'll see. It might be that movie. We'll try it again. And okay, doesn't like that movie. But so what you would have seen if you saw that movie, and that movie was just taken from the Nature paper. But basically, you can see that if you optically activate, let me go back here, escape. So if you optically activate this cell, what you see is that pseudopod extension in that direction. And basically, I just wanted to make the point that the molecular biology is really hard, but the optical activation is super easy. If you want to, we have a microscope stage that will keep your cells nice and happy. If you want to do any kind of optical activation anywhere for any amount of time, it's very easy to do, and it's very intuitive how to do it on these systems. And similar to that, this is the laser capture microdissection system, talking about moving laser beams around. So basically the idea here is this is mostly made for histological type sections, that you place them in general, mostly you place them on these specialized slides that have a little thin piece of plastic on them, and you identify regions of interest. So in this case, breast epithelial cells, and you press go, and it will basically cut out the region that you're interested in and puff it into the Eppendorf tube for further analysis. So this is an example from Joss's lab. His students are interested in the epithelial tissue. They cut out those regions. This is actually imaging the cap right here of the Eppendorf tube. You see that they've picked out those regions. You close up the cap and you send it off to genomics. So this can be done at the single cell level and even the single nuclei level if you're patient enough. So that being said, all the technologies that I talked about so far are limited by diffraction, essentially. And the idea here is if you have an infinitely small point and you image it, you're going to get a point spread function. And I had to get this into a talk, lambda over 2NA. So lambda, the wavelength that you're working with, 2 divided by 2NA, the numerical aperture of your objective. This is going to be the size of your point spread function. If you have a couple of spots within that, they will not be able to be resolved. And just because it's a soapbox of mine, this is actually point spread functions in cross-section. And you can see, so lambda over 2NA, the width of the point spread functions is here, but it's actually the length of the point spread functions that's really dependent on NA, and that's really what changes as you go to lower NA. So super resolution microscopes are microscopes that break the diffraction limit. And this is an example of, a lovely example of chromosomes from Paula Cohen's laboratory. And you can see with normal microscopy, you can't resolve the strands. And this sort of thing, whereas with super resolution microscopy, you can. And this sort of thing is really important to her because she studies chromosome crossover. So where they cross over is critical. So super resolution microscopy is actually a bunch of microscopies with a bunch of very different technologies. This year's Nobel Prize to chemistry went to Betzig, Hell, and Merner for a lot of these technologies. Betzig and Merner were Cornell grads. A lot of you know them, maybe. So the system that we're getting in January can accommodate the first two, the localization-based and the super resolution SIM, structured illumination. Multiple objectives is another way, trying to put as many objectives as you can close to the sample. That helps you. STED, I'm not going to talk about correlative EM. Airy scan is another technology that we're going to be getting in the spring. So localization-based super resolution microscopy is actually, goes by Storm, Palm, GSD. Is actually pretty simple. So if you imagine that these are molecules that you would like to be able to resolve, these little yellow things, if they're within the point spread function, which is indicated by the red blur here, you will not be able to resolve them normally. However, if you can get them to blink, so this comes on and then off, and you basically look for the center of the point spread function, you can identify it. This blinks on, you look for the center, and this blinks on and off, and you look for the center. Now you have resolved your fluorophores to greater than the diffraction limit. And this is an example of Alexa 647 labeled tubulin in a storm buffer. And this is probably not going to play. But it's actually not very interesting. They just blink. And this is the results. A standard image of microtubules would look like this. A storm image then looks like this. So now you've resolved the microtubules. This is taken by members of the Zipfel group. This is an example in onion bulb using this technology, PSF stained cellulose. So the blinking super resolution microscopies. Okay, so these typically result in 20 nanometer resolution, 10 times better than the diffraction limit. So that's great, but they're difficult. They're thin samples. You usually have to use turf or evanescent illumination, which we'll have. They require, the tough part is really the buffers and the fluorophore chemistry. You can only use certain fluorophores. They generally cannot be done live because the buffers are very reducing. And it requires 10,000 images for each super resolution image. So that's sort of like 10 minutes of blinking data, which you guys all missed because I couldn't get it to run. So the other technology that we're going to have is super resolution structured illumination microscopy. So the way that this works is that instead of illuminating uniformly, you illuminate in a diffraction pattern. And then that pattern will beat with the spatial frequencies in your sample. They'll create a beating pattern that's now larger so you can pick it up with your objective lens. So this is how it's implemented in the Zeiss Lyra, the system that we're going to get. So this is actually a three-dimensional diffraction pattern right here. What they do is they basically mix orders of a diffraction grading. And this is an example of an image taken with that. This is a polytene chromosome, and basically looking at heat shock factor, and is in green right here, and polymerase is in red. So you can see that you can really isolate complexes that you can't see in the standard microscope image. So the super resolution structured illumination microscopy, SR-SIM, this is easy. So you basically can put your sample on the stage, and that's it. You can accommodate thicker samples, not real thick, less than 20 microns. It doesn't require specialized fluorophores. It only requires around 15 images, so that corresponds to 5 rotations of this diffraction pattern and 3 translations. So it's relatively fast. It can be done live, and it results in a resolution that's about 2-fold better than diffraction. So there's no such thing as a free lunch. So the Airy scan is going to be kind of similar to the structured illumination microscopy. So this uses a detector array rather than a confocal pinhole in a point scanning laser confocal setup. And this is expected to be about the same as the SR-SIM in terms of resolution and ease of use. It's a really new technology, so there's not a lot of data out there. So we're kind of excited to be getting this in the spring. So the other thing that we have that's new to us is a fluorescence activated cell sorter, or FACS. So if you have suspension cells that you need to separate by color or size, or if you just need to analyze them, this is an instrument that you can use for laser lines, 12 emission colors, specimen cooling. It sorts into tubes or plates, and we can take biosafety level 2 agents if that is of interest. And then other technologies that we have are sort of loosely organized into intravital imaging. And now this can accommodate sort of bigger specimens. They are non-invasive to minimally invasive. So the idea is, a lot of them are set up for live mice, but you could do live plants as well. Plants that are, still in their pot. You'll image them, take them back, then bring them back and image them again if necessary. So this would be luminescence, ultrasound, CT, and in some cases multiphoton. This is an example of our new high-resolution ultrasound. This is a mouse heart beating. So it's good at picking up motion and anatomical structure. It's not used a lot for plants, but I'm not really sure why. This is an image that my daughter took over the weekend. She's 10 years old. So ultrasound is really fast, easy, totally non-invasive, and tissue can be sampled or injected via image-guided needle placement if you would need something like that. Yes, it's Doppler, or it can be depending on how you use it. Visualization and analysis, we support a bunch of different image analysis packages. We do some custom written algorithms. These are our winter analysis, image analysis workshops. If you're interested in finally analyzing all that data that you took, or interested in having your students analyze all the data that you took. So they're going to be this in between break here. So that's all I have.
This has been a production of Cornell University, on the web at cornell.edu.